thank you very much for coming. So, as JD said, today I'm going to give this presentation about the case study that we had in Poplar within an European project, which is WATBIO project, coordinated by Gail Taylor from University of Southampton. Starting from the very beginning, I want to give an introduction on the thoughts that we put on this project, because it was necessary to perform a genotyping-by-sequencing experiment. So, on a large scale with robust data. So, now, if we consider array-based solution, they do are robust assay. Everybody knows that, because chemistries are very intelligent. However, they require such a higher investment, especially for projects that are not considered to go much further during a long time span. And usually those type of assays requires a minimum order, which usually for the most famous companies is in the order of several hundred of samples. Also, those arrays are some limits in the throughput. So the number of polymorphisms that can be assayed are in short ranges. And also, the most important thing is that those assays, given research in the context of research projects, they put ascertainment bias in whatever diversity you are sampling. So if a panel of diversity has been discovered in a given germplasm, that doesn't mean that that assay then is going to work greatly in another panel of diversity. On the other hand, we have NGS technologies that we are seeing keep decreasing the cost. They provide good scalability with several sequencing machines, and also with new engineered kits that provide many barcodes to higher multiplexing. Then everything is sequence-based, so there is sequence information which can be used for other purposes, not only for the genotyping scope. And also potentially it's an ascertainment bias-free information. So we have seen technology improvements, that is the read length is keep increasing, so there is much more information to leverage, and also for many genomes now the accessibility to have a draft genome have became an easy reality. However, we still have some challenges in the efficiencies of performing a very educated genotyping by sequencing experiment. Some of them are genome complexity, optimization of the multiplexing, as I said before. Also, maximization of the calling ratio, because many people have performed genotyping by sequencing, but I heard that not many were at the very end happy about the calling ratio. And also, to maintain in the system an adequate coverage when dealing with heterozygous and polyploid genomes. So I think we all are aware about genotyping by sequencing in the early meaning of the term, which is performed by using restriction enzyme to reduce the complexity of a genome. There are several fashion of protocols, some relying on a single enzyme, which is some of a shotgun approach more or less, and maybe more sophisticated protocols relying on 2 enzymes and site selection, which can provide more fine-tuning on the complexity reduction by limiting the number of loci. They work very well, but we might have some limitation. I will tell about this later in my slides. So the complexity reduction must be precise to have, let's say, an happy genotyping, so where the cost effectiveness of the experiments are well tuned. And we might have several scenarios of study. We can go from phylogeny to population structure and linkage analysis, and then up to association studies. And what there are, depending on the type of study that we are carrying, there are the minimum amount of markers is driven by different factors. In case of phylogeny, divergence is the limiting driver, while for linkage analysis, recombination is our limiting factor through the saturation of the data point that are necessary for the complete analysis. While in association studies, we have linkage disequilibrium playing as a limiting factor. So, on top of this, the project was to perform a genome-wide association studies in Populus nigra. So we need a massive genotyping actually, this is wrong, it's 540, sorry, accession at the very end of the project. And we want to reside on the gene space because it's an outcross species, so the linkage decay is very rapid. So the idea to perform a random shotgun genotyping by sequencing was not very our best solution in our mind, because we might have, yes, many markers, but likely not in very strong linkage with the causative mutation of a given trait of interest. And we had these resources. So we have low-pass sequencing, that is some, let's say, 2x to 10x coverage of 51 accessions of Populus nigra. And we also have the Populus trichocarpa genome sequence available. And also, for benchmarking purposes, we can rely on some Illumina Golden Gate data that was generated in a previous study. So, how the SPET technology was originally designed by NuGEN. So, the original paradigm was to have some target region of interest, most of the cases talking about exons. The probes that are not real probes, actually those are kind of a primer, are designed on flanking regions of such target site. And the fragments that are generated from the library preparation are sequenced on a single-end fashion to get some sequencing on top of the target region, scattered across itself by means of the standard deviation of the fragmentation on the library. So we thought, okay, no, sorry, another, I think I will skip this one. No, not really, I'm not going to skip this because JD doesn't say this. So this is the final library product where we have a forward primer and a reverse primer. The reverse primer originally was attached to the probe sequence, and that was not going to be sequenced in a standard fashion. While on the other side we have the forward read, which is randomly distributed across the region, and then the index primer will sequence 8 nucleotides, which works as a barcode, and 6 nucleotides, which work as they are random exomers. I think the newer kits have 8 bases, and those are used for the duplication of PCR duplicates. So we thought, okay, why not to use this system the way around? So to design a probe just nearby a SNP site of interest, in order to have all the sequences coming from the sequencing of the reverse read, because this is the way we did it the very first time, to maximize the coverage on top of the target SNP. Well, on the other end, we also performed the parent sequencing, so the other reads were also coming along, providing new SNP discovery and genotyping in the nearby region. One caveat was that was a very highly heterozygous species. So luckily we had the low-pass sequencing from the 51 accessions of Populus trichocarpa, so we had a database of known polymorphisms, or at least the most common ones. And so we asked NuGEN to design probes in order also not only to be on the right distance with respect to the target SNP, but also to avoid to land on other non-SNP sites, and this is to prevent allele-specific amplification. So this paradigm was looking to maximize the sequencing output on the selected polymorphism, have a genotyping system which is duplicate aware, and also to integrate a target approach and a random, let's call it de novo genotyping approach. So how we design the probes, we select regions on a priority rank from exon down to up and downstream region of the genes. We impose a rule to have an average of 3 SNPs per gene, but actually depending on the gene length, we scale from 1 SNP site to 4 SNP sites per gene. Sites had to be at least 500 base pair apart to don't be redundant in linkage disequilibrium. The minor allele frequency to be higher than 0.15, and we select a candidate panel of 110,000 SNP sites. So before investing a lot of money, we did a test panel with just 8 samples and just 12,000 probes out of the big panel, and we did sequence 1 to 2 million reads per sample. I want you to keep in mind the allele frequency distribution coming out of our selection of the SNPs, given the low-pass sequencing data that we had in our hand. The very first good thing we saw out of the sequencing is that the enrichment efficiency was very high. In our lab, we do a lot of enrichment capture experiments, and the hybridization-based one on non-model species, if you are lucky, you get 50, 60, at the maximum 70, but I don't think so. So I would say 60% enrichment efficiency is what you get, especially on non-model species and on very small target regions. But here we saw an average of 87% of enrichment efficiency. Is this my left time?
Correct.
Oh, just okay. I thought it was more.
You can take more.
Oh, okay.
It's just a visual.
So we look at the distribution of coverage across all the loci. What you can see here is that a comparison between 2 samples shows that yes, we do have a variance across loci, but that variance is reproducible. Those are kind of over-sequenced loci, but there are not so many. So the most of the juice is here, where we can do genotyping, and those that are poorly covered are somewhat reproducible. And so that does means that yes, you can keep in mind that some loci might be lost, but those are the same loci. So there is no combinatorial effect like in the standard genotyping by sequencing, where the coverage drop is random, and so the more the sample you add to the system, the more the calling ratio will drop. We also, through another experiment, look at if it was a good idea to use not only 1 probe per SNP site, but 2 or even 4, so 2 on both sides. What we saw is that yes, you can reduce the amount of probes with 0 coverage at the very end, but the distribution of the coverage was not. I was expecting actually something better, but the increase in the efficiency of the coverage per probe was not so very high. So the final outcome of this test was, okay, it's not worth to invest more than 1 probe per SNP site. Just accept that some of them will be maybe lost, but it's better to use a single probe per SNP site. Probably this is due to some regions of the genome that were poorly characterized, so we didn't know the presence of other polymorphisms or structural variation, so those were hampering the primer extension anyhow. So no matter if you add more primer probes on that region. So we test our data against a known data set of samples that were genotyped using the Golden Gate assay. And we only had 6 samples by a matter of some 100 overlapping SNP loci. And what we saw that actually the recall ratio against the Golden Gate data was just a matter of coverage. So you see that with the mean on-target coverage going up, you basically have 100% recall rate against the Golden Gate. Yes, of course, this is data based on just 6 samples, but it does gave us a clue that the system was not introducing a bias or a systematic error in the genotyping efficiency. So we did some simulation with this data to project what would be the probability of having missing data or false homozygous calling given several sequencing layouts for the big project for the 90,000 SNP panel. And so, given our simulation, we decided to stick with the 48-plex sequencing on a high-seq 2500 lanes. And we went ahead by sequencing the full set of genotypes, 540 540 samples with a kit of 90,000 loci, and the target was to sequence some 8 million suites per sample. So this is the distribution of the sequencing yields we had across the samples. You can see that it's pretty normal. We have at the very end we had a median of 9.5 millions of reads per sample. And to accomplish this normal distribution, we adopt, let's say, resorting exercise on plates. So when we get the DNA, the samples in plates, we quantify after the sonication step that now probably is not going to be necessary any longer given the enzymatic digestion, and with the values of DNA concentration, we resort to the plate with the robot to have the samples that are going to be pulled together and subjected to the enrichment step on the same pool. So higher concentration sample together, lower concentration samples together to homogenize the efficiency, given the fact that each single pool is going to be through a further quantification step to be loaded on the sequencer at the very end. So this is the analysis pipeline. We use the regular, of course, the Cassava software to perform the multiplexing out of the Illumina machine. So we adopt a SPET-specific trimming procedure because the adapters include some base offset to augment the complexity of the reads. We perform alignments with BWMM, and then we remove the duplicates. And for the records, we saw that the average duplication rate was around 10%. SNP calling was performed with the GATK suite. Then we apply some SNP filtering and we mask the probe regions. And at the very end, we end up with some 10 million row SNP sites, which of course are a lot. Probably they are true, but we want to only look at those that have a very high genotyping ratio. So we apply some criteria to filter for, and we end up that out of the original 90,000 SNP panel, we got genotypes for 66,000 sites. But not only, by leveraging the extra sequencing that we got out of the pair reads or the reminder sequence that we got from the reverse read, we further genotype other 453,000 and more SNP sites. And this, as JD showed right before, is the typical profile of a SPET library sequence on pair end, which means that you have a stack of reads just opposed to the probe site, where you can genotype your site of interest, and then you get something more for free either from here or from here. I think that, and at the time we did this experiment, for us it was mandatory to do the parent sequencing because the kit that we're providing was having the probe on the reverse read side. Now they're providing kits with the probe on the forward read. So single-end sequencing is enough to have the stack of reads to maximize the coverage if you want to save money. Do you remember the profile of allele frequencies? Okay, here is the profile we got out of our genotyping experiment from the panel out of the SNP. From the SNP out of the panel. And here from the SNP that were genotyped de novo. So this shows that the system, given not, since it's not totally target, can yield rare alleles. And this is a principal component analysis, either made by the SNP that were in the original panel and the SNP that have been sampled from other sites. And you can see here at the center there are population of Populus nigra which are hardly distinguishable, because the problem is the ascertainment bias, because we had the set of samples from which we discovered the SNP site was skewed for the presence of French, I think it's French and Spanish genotypes of Populus nigra. While here, when using also new SNP sites, we can have a much better discrimination of groups. Like a clear example is here, and compared to here, where this gray group overlap with this fancy colored group. So the project was, all of this was to perform a genome-wide association study. And this was carried by Michael Wright from University of Southampton. This is the pipeline he used for performing the analysis. I'm not going to be in the details. He filtered the SNPs by linkage disequilibrium, neutrality. He performed some analysis of the structure of the population. And at the very end, he conducted some tests for association. And I just want to show you that the system was indeed successful to retrieve candidate SNP sites involved in traits of interest. In this slide, I'm showing you the significant polymorphisms that are associated with leaf sites on 2 independent data sets. And they are, all of them are recalled on both of the samples, on both of the data sets. So I want to conclude that this system can both genotype target SNPs and discover novel polymorphisms. So it's a very promising strategy if you want to do fine mapping. You have to deal with unassembled complex genomes, but at least you require some transcriptome. Very good for genome-wide association studies, or for low diversity genomes, where regular genotyping-by-sequencing methods, which are sampling the genome randomly, hardly will pick SNPs, while if you rely on known database of polymorphisms, you can have a much more cost-effective approach here. This is an old slide, because here it's more expensive than GWAS RAD. Well, with what JD just showed before, I don't think this is any more true. I think it's comparable to the cost that you can get from GWAS RAD provider like us. And the probe synthesis can scale well. Actually, I wasn't aware that now they are providing also probe set lower than 5,000 probes. As I said, we try to use more probe per SNP site, but it doesn't really pay off. And also, as JD said, you can run a test sample on some SNP site, then you can increase the panel of SNP sites, putting more probes, or like we want to do in other projects, since we want to better homogenize the coverage, you can start with a test panel with many more SNP sites, and then just choose those that are working at the best to have a higher scale experiment. So I thought I had more time, so I put some extra slides, but I think I'm going to skip the Allelic-specific. We also use this system to validate Allelic-specific expression using the SPET system on cDNA. And I'm not going through this because I'm late in time, but I think this.
You have some time if you want to go.
Okay, okay. So just 5 minutes. So allelic-specific is that phenomenon where the two alleles on a diploid genomes are not expressing the same gene at the same level. We can have a balanced configuration where both orthologs, sorry, not both homologs are expressed at the same level. But in some cases, we can have genes that are one allele is not expressed, or they are highly unbalanced. Or we can have we can have loss of expression and other scenarios. And what is causing this can be epigenetic modification, structural variation, polymorphism in the promoter region or the enhancer region, transacting elements, and also nonsense mediated decay. And in our lab, we are studying this phenomenon in the context of structural variation of a great genome. So we use RNA-Seq as a primary source of evidence to see if there was allelic-specific expression. And we utilize Allelic software, which is a software where you provide one reference and an alternative reference as representative of an alternative haplotypes. It does some permutation and correction of the data to provide some results on whether a given gene has unbalanced allele-specific expression given the presence of some polymorphism on the coding sequence. And we get some genes that were significant for allelic-specific expression. So we said, okay, why not to try a SPET technology as an independent method to validate those results? So we apply the same paradigm of genotyping, but on cDNA, targeting SNPs that were involved in genes that show allelic-specific expression given the former analysis. So we filter out the SNPs that were satisfy certain criteria, such as the presence of at least one read, to have an allele frequency between 0.4 and 0.6 on a test and a control made from gDNA, and also to already being significant from the former test. And by filtering those sites, we got this. So you can see how, and in the x-axis we have the log2 ratio from RNA-seq data, and on the y-axis we have the log2 ratio from the cDNA-SPET log data, and we can see how they correlate. So the SPET method show that can be also used to quantify allelic-specific expression, and probably in the future experiment we can use it directly to assess the presence of allelic-specific expression. Just want to note that on this graph you can see for RNA-seq data the software provides a value for a single exon, while on cDNA-SPET experiment we sample several SNP for a given exon, and that's why on the same vertical axis we can see several dots which are like this one. This is a single exon out of RNA-seq data, compared to several SNP sites out of the cDNA-SPET data. And you can see that, yes, they go up and down, but on average, they are in line with the RNA-seq log2 ratio. So I want to conclude saying that SPET can also be used as a proxy to cost-effectively screen allelic-specific expression, and especially can be very useful in low-diversity species. I want to thank all the contributors to this work of these works from IGATEC, Federica Cattonaro, who is the CEO, Eleonora Di Centa, who prepared the libraries in the lab, and Simone Scalabrin, people from University of Southampton, Mike, Wright, Gail Taylor, and Hazel. They are in charge of the Whole Genome Association study, and also people from the IGA, Professor Michele Morgante, Fabio Marroni, Mara, and Eleonora for the allelic-specific part. And thank you all for the attention and for the patience. Thank you.
One or two questions, maybe?